Hello, and welcome to an exciting journey through a revolutionary advancement in medical science, nanoparticles for lung gene editing. In recent years, the integration of nanotechnology with genetic medicine has opened new vistas for treating diseases that were once considered untreatable. This essay delves into how nanoparticles are poised to transform the landscape of gene editing, particularly in treating lung-related conditions. The fusion of these two cutting-edge fields holds the promise of not only treating but potentially curing a range of genetic disorders that affect the lungs. Gene editing, a method that allows scientists to alter an organism's DNA, has shown immense potential in curing genetic disorders. Techniques like CRISPR have revolutionized our ability to make precise changes to the genome, offering hope for conditions that were previously deemed incurable. Coupled with the precision of nanotechnology, this approach can target lung cells more effectively and safely than ever before. Nanoparticles act as tiny delivery vehicles, carrying genetic material directly to the cells that need it most, minimizing side effects and maximizing therapeutic benefits. Nanoparticles, due to their minuscule size, can navigate the complex environment of the human body, especially the intricate network of the lungs, to deliver genetic modifications directly to the affected cells. This precision targeting is crucial for treating lung diseases, where traditional methods often fall short. The potential of nanoparticles in lung gene editing is vast, offering hope for conditions like cystic fibrosis, lung cancer, and even chronic obstructive pulmonary disease, COPD. These diseases have long been challenging to treat due to the difficulty in delivering therapeutic agents directly to the lung tissue. As we move forward, you'll gain a clearer understanding of how these tiny particles work and why they could represent a major leap forward in medical technology. We'll explore the mechanisms by which nanoparticles can be engineered to carry specific genetic instructions and how they interact with lung cells to bring about desired changes. By the end of this essay, the significance of nanoparticles in lung gene editing will be both clear and compelling. You'll see how this technology is not just a theoretical concept but is already being applied in research and clinical trials, showing promising results. In this section, we introduced the transformative potential of nanoparticles in the realm of lung gene editing. The ability to precisely target and modify genes within lung cells opens up new possibilities for treating a range of pulmonary conditions. As we progress, we'll explore more about gene editing techniques and the specific role of CRISPR technology in this field. CRISPR, with its ability to make precise cuts in the DNA, is a powerful tool that, when combined with nanoparticles, can enhance the efficiency and accuracy of gene editing. The journey into this cutting-edge science promises to be as enlightening as it is promising. We'll delve into the latest research, discuss the challenges and ethical considerations, and look at the future prospects of this exciting field. Stay with us as we uncover the fascinating world of nanoparticles and gene editing, and discover how these innovations are set to revolutionize the future of medicine. The potential to change lives and improve health outcomes is immense, and we are just at the beginning of this incredible journey. Gene editing is a groundbreaking scientific technique that involves altering the DNA sequences of living organisms. This revolutionary approach allows scientists to make precise changes to the genetic material, opening up a world of possibilities in medicine, agriculture, and beyond. This can be used to correct mutations that cause disease, enhance certain biological capabilities, or even create resistance to viruses. By targeting specific genes, researchers can potentially eliminate genetic disorders, improve crop resilience, and develop new treatments for infectious diseases. One of the most significant advancements in gene editing has been the development of CRISPR-Cas9, a technology that has streamlined the process of genetic modification. This tool has revolutionized the field by making gene editing more accessible and efficient. CRISPR, clustered regularly interspaced short palindromic repeats technology, uses a protein called Cas9 as a kind of molecular scissors to cut DNA at a specific location. This precise cutting allows scientists to target and modify genes with unprecedented accuracy. When this cut is made, the cell's natural repair mechanisms kick in and allow for the addition or removal of pieces of DNA. This process can be harnessed to insert new genetic material or delete faulty sequences, paving the way for innovative treatments and therapies. This method is highly precise, efficient, and less costly compared to previous techniques, making it a revolutionary tool in genetic research and therapy. The ability to edit genes with such precision has far-reaching implications for various fields. including medicine, agriculture, and environmental science. In the context of lung diseases, CRISPR offers a promising pathway for creating targeted therapies that could potentially correct genetic mutations at their source. This approach could lead to more effective treatments with fewer side effects, improving the quality of life for patients. For example, in conditions like cystic fibrosis, where a single gene mutation leads to severe respiratory symptoms, CRISPR can be used to directly modify the faulty gene in lung cells, potentially reversing the disease's effects. This could transform the way we approach genetic disorders, offering hope for a cure. This image illustrates how CRISPR targets and modifies DNA within lung cells, symbolizing a significant stride towards personalized medicine. Personalized medicine aims to tailor treatments to individual patients based on their genetic makeup, and CRISPR is a key tool in making this vision a reality. As we proceed, we will explore how nanoparticles make the delivery of CRISPR tools to the lungs more efficient, further enhancing the potential of gene editing for pulmonary diseases. Nanoparticles can protect the CRISPR components as they travel through the body, ensuring they reach their target cells and perform the desired genetic modifications effectively. Delivering genetic material to the lungs presents unique challenges due to the organ's complex structure and protective mechanisms. Traditional methods, such as viral vectors, have been used to deliver genetic material, but often come with risks of immune responses or other complications. This is where nanoparticles offer a groundbreaking alternative. Nanoparticles can be engineered with surface properties that allow them to bypass the body's immune surveillance and deliver genetic editors like CRISPR directly to lung cells. These particles are designed to be small enough to navigate through the bloodstream and into the lungs' alveoli, the tiny air sacs where gas exchange occurs. Their size and surface characteristics can be meticulously controlled to optimize delivery and minimize potential side effects. For instance, researchers can modify the surface of nanoparticles to avoid detection by the immune system, ensuring that they reach their target cells without being intercepted. Additionally, these particles can be equipped with special ligands that bind specifically to receptors on lung cells, further enhancing the precision of delivery. This image captures the concept of a nanoparticle engineered for lung delivery, highlighting its potential to revolutionize lung therapy. In the next section, we will delve deeper into how these nanoparticles are specifically used for gene editing in lung cells, marking a critical advancement in pulmonary medicine. The integration of nanoparticles with gene editing technologies, especially CRISPR, is a significant innovation in the field of lung therapy. This groundbreaking approach combines the precision of CRISPR with the advanced delivery capabilities of nanoparticles, creating a powerful tool for treating lung diseases at the genetic level. These nanoparticles act as vehicles that carry CRISPR components directly to the affected lung cells, ensuring that the gene editing tools do their work precisely where needed. By targeting specific cells, this method enhances the accuracy of gene editing, reducing the likelihood of unintended genetic modifications. This targeted approach not only increases the effectiveness of the treatment, but also significantly reduces the risk of off-target effects, which are a major concern with traditional gene therapies. Off-target effects can lead to unintended consequences, making precision a critical factor in the success of gene editing therapies. Nanoparticles are designed to be biocompatible and degrade after their payload is delivered, minimizing long-term exposure and potential side effects. This biocompatibility ensures that the nanoparticles do not provoke an immune response, which could complicate the treatment process. This transient nature of nanoparticles is crucial for safe clinical applications, especially in sensitive organ systems like the lungs. The ability to degrade and be cleared from the body reduces the risk of chronic inflammation or other adverse reactions. Moreover, the ability to control the time and place of CRISPR delivery enhances the efficacy of the treatment, allowing for controlled gene editing that is synchronized with the body's natural cycles. This synchronization can be particularly important in ensuring that the gene editing occurs at the optimal time for cell repair and regeneration. The development of nanoparticle-based lung gene editing is also facilitated by advancements in imaging and nanotechnology. These advancements allow scientists to visualize and track the nanoparticles as they move through the body, providing valuable insights into their behavior and effectiveness. Researchers can track the movement and distribution of nanoparticles in real time, ensuring that they reach the intended destination and perform as expected. This real-time monitoring is essential for adjusting the treatment protocol as needed to achieve the best possible outcomes. This level of control is pivotal in developing therapies that are both effective and safe. By fine-tuning the delivery and activity of CRISPR components, researchers can maximize therapeutic benefits while minimizing potential risks. This detailed illustration shows nanoparticles at work, delivering CRISPR components into a lung cell. The visualization helps to understand the intricate process of gene editing at a cellular level, highlighting the precision and efficiency of this method. The precision and efficiency of this method could drastically change how we approach genetic disorders of the lungs. By directly targeting the genetic root of these diseases, we can develop more effective treatments that address the underlying causes rather than just the symptoms. As we continue, we will explore the specific diseases that could be treated using this technology and the potential for personalized medicine. Personalized medicine tailors treatments to the individual genetic makeup of each patient, offering the promise of more effective and customized healthcare solutions. This approach could revolutionize the treatment of lung diseases, providing new hope for patients with conditions that were previously difficult to manage.